Good morning. It's January 17th, and this is your daily brief in science. Here's everything you need to know. A recent study has highlighted a concerning link between COVID-19 and myalgic encephalomyelitis/chronic fatigue syndrome, or ME/CFS. Individuals who contracted COVID-19 are facing a fourfold increase in the risk of developing ME/CFS compared to those who remained uninfected. Among participants diagnosed with post-COVID-19 ME/CFS, a striking 89% were also identified as having long COVID, indicating a significant overlap between these two conditions. Dr. Suzanne D. Vernon, the lead author of the study, emphasizes the urgent need for healthcare providers to recognize ME/CFS as a diagnosable and treatable condition that can follow COVID-19. This research is part of the NIH's Recover initiative, which seeks to enhance understanding of long COVID and its associated conditions through collaboration with various stakeholders, including clinicians and patients. The study analyzed data from over 11,000 COVID-19 patients, focusing on the incidence of chronic fatigue syndrome post-infection. It revealed concerning links between long COVID symptoms and chronic fatigue syndrome, characterized by debilitating symptoms such as chronic fatigue, cognitive impairment, and unrefreshing sleep. Potential causes of long COVID may include autoimmune reactions, reactivation of dormant viruses, or damage to the vagus nerve and brain communication pathways. The authors suggest that the reported 4.5% incidence of CFS is likely an underestimate, as hospitalized patients were excluded from the analysis. However, the study acknowledged limitations, including reliance on self-reported symptoms and the exclusion of hospitalized COVID-19 patients, which complicates the diagnosis of ME/CFS. Additionally, the study found that older age, female sex, and pre-existing pain conditions were associated with a higher risk of developing ME/CFS following COVID-19 infection. Deep ocean ecosystems remain one of the least understood areas of our planet, and a new tool aims to change that. DeepSea, a collaborative visualization system developed by researchers from the Georgia Institute of Technology and California Institute of Technology, is designed to enhance the analysis of these complex environments. DeepSea features an interactive workspace that integrates three coordinated views: map view, core view, and interpolation view. This setup facilitates comprehensive data analysis, addressing critical design challenges related to effective data integration and analysis, which are essential for studying deep ocean ecosystems. The remote nature of deep ocean research, coupled with high sampling costs, necessitates efficient decision-making tools for scientists. Findings indicate that visualization tools like DeepSea can significantly enhance decision-making capabilities and foster a deeper understanding of complex scientific processes. With DeepSea, scientists can visualize both 2D and 3D interpolations of data, which aids in identifying optimal sampling locations and maximizing scientific returns. In the map view, users can annotate maps with drawings and notes, supporting the visualization of sampling locations. The integration of various data types, including core data and environmental metadata, is crucial for contextualizing findings. DeepSea promotes fluid interaction and rapid data integration, essential for on-the-fly decision-making during deep ocean fieldwork. Initial field tests aboard a research cruise demonstrated that DeepSea increased scientific returns, catalyzed new research workflows, and improved communication among team members. Expert interviews highlighted the importance of user feedback in refining DeepSea and enhancing its effectiveness. Additionally, the system's design includes colorblind-friendly palettes for visual accessibility and allows users to deselect cores for focused analysis in the interpolation view. NASA's upcoming Pandora mission is set to revolutionize our understanding of exoplanets by analyzing their atmospheres. This marks a significant advancement in astronomy since the discovery of exoplanets in the 1990s. Astronomers will utilize transit measurements, observing how light interacts with atmospheric substances as planets pass in front of their stars. Over the course of a year, Pandora will observe each of its 20 targets 10 times, with each observation lasting 24 hours and focusing on transit measurements. The mission aims to study at least 20 known exoplanets, concentrating on identifying key atmospheric components such as hazes, clouds, and the presence of water. The concept for Pandora originated in 2018 when researcher Benjamin Rackham identified challenges in distinguishing starlight from exoplanet signals, which could limit the capabilities of the James Webb Space Telescope. Designed to observe exoplanets for longer durations than Webb, Pandora will provide deeper insights into their host stars and improve clarity regarding planetary atmospheres. Equipped with a 45-centimeter aluminum telescope developed by Lawrence Livermore National Laboratory and Corning Specialty Materials, Pandora will capture brightness and near-infrared spectra of both stars and exoplanets. The project is managed by NASA's Goddard Space Flight Center and involves significant contributions from Lawrence Livermore National Laboratory, Corning, and Blue Canyon Technologies. The mission is expected to enhance our understanding of exoplanets, including potentially habitable worlds, thereby expanding the search for life beyond Earth. Data collected by Pandora will also aid in interpreting measurements from the James Webb Space Telescope, which is similarly designed to search for habitable worlds. The announcement of the Pandora mission was made at the 245th meeting of the American Astronomical Society in National Harbor, Maryland. Carl Harshman, leading the mission operations team at the University of Arizona, expressed readiness for the launch and excitement for the scientific data that will follow. A groundbreaking study has emerged that could change the landscape of Huntington's disease research. Published in the journal Cell on January 16, 2025, this research sheds light on the mechanisms behind this hereditary disorder, which causes severe movement, cognitive, and behavioral issues. Conducted by a team from the Broad Institute of MIT and Harvard Medical School, and McLean Hospital, the study identifies how the Huntington's mutation leads to brain cell death. The mutation stems from an inherited alteration in the Huntington gene, characterized by a CAG DNA sequence that repeats at least 40 times. Researchers analyzed brain tissue from 53 Huntington's patients and 50 control subjects, revealing that striatal neurons exhibited repeat tracks with up to 800 CAGs. They found that significant gene expression distortion and cell death occur once these repeats exceed approximately 150. Notably, the somatic expansion of CAG repeats was found to happen specifically in striatal projection neurons, leading to cell death only after surpassing a critical threshold. The study indicates that these repeat tracks grow slowly during the first 2 decades of life, but then accelerate significantly after reaching around 80 CAG repeats. This suggests that intervening to stop or slow down CAG repeat expansion could delay or even prevent the onset of Huntington's disease. The research team emphasized the potential for new therapeutic strategies that focus on slowing DNA repeat expansion rather than just targeting Huntington protein expression. Furthermore, the findings may have implications for other genetic disorders caused by DNA repeat expansions, such as Fragile X syndrome and myotonic dystrophy. Dr. Mark Mailer referred to the study as landmark, addressing long-standing questions about the late onset of Huntington's despite the early presence of the genetic mutation. Currently, about 41,000 Americans are affected by Huntington's disease, with available treatments primarily managing symptoms rather than providing a cure. The challenges faced by recent trials of experimental drugs aimed at lowering the toxic protein have prompted researchers to consider DNA repeat expansion as a new target for future treatments. A recent study from the University of Notre Dame has raised significant concerns over the presence of toxic PFAS, or forever chemicals, in smartwatch and fitness tracker bands, which can be absorbed through the skin. This research tested 22 common brands and discovered that 15 of them contained high levels of PFAS. Alarmingly, 9 bands exhibited dangerously elevated concentrations of PFHXA, a synthetic PFAS associated with various health issues. Health risks linked to PFAS exposure include several types of cancer, liver and thyroid disorders, and immune suppression, underscoring the hazards these chemicals pose. The study revealed that PFHXA was detected in 40% of the bands tested, prompting calls for further research into its potential health implications. Co-author Graham Peasley highlighted that some samples contained PFHXA levels exceeding 1,000 parts per billion, a concerning threshold for consumer products. While the study did not name specific brands, it indicated that popular names like Nike, Apple, Fitbit, and Google were part of the analysis. Interestingly, elevated PFHXA levels were more common in pricier watch bands, particularly those costing over $15, with some bands exceeding $30 showing high fluorine content. Researchers suggest that consumers might consider silicone bands as a safer alternative to those containing PFAS, recommending avoidance of these harmful chemicals when feasible. Despite the growing body of evidence regarding the dangers of PFAS, regulatory actions in North America have been limited, with only a handful of states taking measures against these chemicals in consumer products. In contrast, Europe is advancing towards stricter regulations, with the EU proposing a ban on PFHXA in consumer goods, reflecting an increasing scrutiny of PFAS. This study marks the first investigation specifically focused on PFAS in wearable technology, highlighting an area that has previously received little attention. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.